Welcome to DaSL, the podcast from the Data Science Lab in the Johns Hopkins Department of Biostatistics. My name is Brian Caffo. I want to remind everyone that opinions stated and expressed from this podcast are from the host and guest alone and not that of the Department of Biostatistics, Johns Hopkins University, etc., etc. So I'm delighted today to be joined by Ciprian Crainiceanu, the Myrto Lefkopoulou, the second Myrto Lefkopoulou Award winner on the podcast. Ciprian is an extremely noted both methodological and applied biostatistician. I think he's one of the best biostatisticians in the world. He was the creator of the SMART group. He has published in an incredibly large spectrum of methodological and analytic areas, and he's definitely my go-to person. If I ever have a question about statistics, the first person I go to is Ciprian, who is also my number one collaborator. If I look at who I've worked with the most, it's Ciprian. He's just a lovely person to work with and a great friend. So welcome to the podcast. Thank you for coming.
Thank you for having me, Brian. I'm absolutely delighted to be here. And you just told me, hey, do you want to do this? And I'm like, sure. I don't know what it is, but we'll do it.
And we usually save pitching stuff till the end. But you might notice in the background, we have, Ciprian has a new book out called Functional Data Analysis with R. It's really an impressive book. It's with friends of the podcast who will be on there as well. We hope to get his guests soon, Jeff Goldsmith, Andrew LaRue, and Erja Cui. It's really a wonderful book because just like how I described Ciprian, it has this wonderful blend of teaching you how to do things, so it actually has coding in R, but also the sort of why and the conceptual foundation of the thing. So it's really a nice book. Anyway, if you're interested in functional data analysis, especially if you want to do functional data analysis, if you want to, at the end of the day, have some kind of analytic product out, then this is the right book for you.
Thank you so much. I'm really excited to have written this book with former students and colleagues, and it also has a website. So if you're interested in that, you can get the data sets. Everything is reproducible, and we love it.
Awesome. Well, that's the pitch part of the podcast. We're hoping to get like a mattress company to sponsor us next. So I wanted to start. So Ciprian graduated from his PhD from Rutgers, working with Dave Rupert, and
Cornell.
Yeah, and I don't even know where I pulled Rutgers from. Yeah, from Cornell.
That's Martin.
Yeah, that's Martin. That's right. Anyway, working with Dave Rupert, and you worked on a problem that was kind of near and dear to this department. So I'm thinking, of course, about the zero variance component testing problem. I wonder if you could talk a little bit about that. Maybe mention a little bit of the history with Self and Liang here.
Yeah, it's one of these problems, right? I mean, you do statistics, and all of a sudden you work on different problems, and you realize that many of these problems have something in common. For example, you work on linear mixed effects models, and you want to see whether there is heterogeneity in the data, or you work on smoothing and you want to see whether the model is parametric or not. And then you realize that all of these problems have something in common, and that's testing for variance component. And this was not a new problem that I worked on. Professor Liang, who was here in the department and one of the best known statisticians in the world, worked on it. And when I started working on it, the problem was thought to be solved in some sense. And the only problem was that when we were starting to simulate in different contexts these results, we were not getting exactly the theoretical things. And we thought maybe it was our problem, maybe the simulations were wrong, but the more we did those simulations, the more we realized that something was different. And my advisor told me about it. He showed me his simulations. I redid them, and then there were a few other results in the literature that said Yeah, it's not exactly what people think. And then in the end, what I did was to realize that the main result that was at the time thought to apply everywhere was based on an idea of independence of vectors, and that's not always the case in many applications, and that kind of illustrated the theoretical part of it, and then we apply that in different contexts, and it's been one of these problems that over time, just kept on going, and it's still in many ways an open problem, and on the other hand.
So wait, and you did you corrected the asymptotics and did the finite sample distribution.
The finite sample distribution, yeah, and it's one of these problems that keeps on giving. We even now, many years later, we encounter that, and you encounter it in various areas in genomics when you test for heterogeneity. Everywhere where there is a mixed effects model, there is a question: is the variance zero or not? And that relates directly to a scientific problem. It's not just an abstract idea that, let's make things hard and test for variance components.
And one thing I learned from you is how that problem connects to a seemingly unrelated problem, which is the scatter plot smoothing. That scatter plot smoothing is at some level a zero variance component testing problem. Can you comment on that?
Yeah, it's one of these things that is unexpected and interesting at the same time. I think it also goes back to a philosophical problem, which is testing whether you have a parametric versus a non-parametric model. So, and in scatter plot smoothing, essentially you can show that, and this was shown not by us, by many illustrious statisticians, that scatter plot smoothing can be seen as standard regression with a penalty, and that penalty is directly related to a variance component. And at the limit, testing whether you have a parametric versus a non-parametric can be shown to be testing for a zero variance component. And this is something that it was kind of obvious, but I think it came from all the work that Grace Waba did, and then many others, including Marx, Eilers, and Rupert, and all. And I think I was among the first ones to identify that the problem was testing for a zero variance component. The difference was that in scatter plot smoothing, you don't have multiple vectors, so the data doesn't parse out in independent vectors as it does, for example, in standard multi-level models. I hope I don't get too technical here, but that's essentially the.
Yeah, no, it's totally okay. Yeah. Okay, I see. So the fact that, like, in the Self and Liang work, it was sort of IID chunks from longitudinal data analysis is what made it work. But then when you moved it to a testing for a non-parametric component, you lost some of those independence asymptotics in that sense.
Exactly. So the Self and Liang is a correct result under the assumption that the number of groups goes to infinity.
Yeah.
And the problem is that in many situations, the number of groups doesn't go to infinity, and it's small or moderate in size, and then all of these problems occur.
Nice. And if you want to learn more about this stuff, you can read it in the functional data analysis book. So, which brings us to kind of the next topic that you really spent a significant chunk of time on, is functional data analysis. I don't know if you can maybe talk a little bit about functional data analysis and.
Well, I can blame you for that. I mean, I can blame you for many things, including what some consider to be success in my career. So it all started a long time ago. I can't believe I say that, but it was a long time ago. And you walked into my office and you introduced me to Naresh Punjabi, a very dear collaborator now. And he was working on EEG data, and we were trying to understand the patterns of signals in the human brain. And essentially the study was that somebody was hooked to an EEG machine over the course of the night, and you have this very high resolution data, and you try to make sense of it. And then functional data analysis, which is essentially the analysis of functions, was a very natural way to think about, where the function was the continuous measurement of the brain activity during the night. And then we realized that many other medical and epidemiological problems share a similar structure. The methods are not always the same, they vary, they are different from one application to another, but that's how, I kind of had to go to that literature. And yeah, I mean, that introduction that you made, and then our collaboration over the years, and that changed my career.
This is 20 years ago.
20 years ago.
It was 20 years ago, actually. Now that I think about it. Yeah, because you joined the department in like 2003?
2004 I started.
2004.
Yeah.
Okay, so yeah, 20 years ago.
20 years ago, exactly.
After working on functional data analysis, you applied these techniques and others in the field of magnetic resonance imaging, and especially as it applies to multiple sclerosis. And I'm wondering if you could maybe mention a little bit about your work in MRI and MS. And
yeah, it's been a privilege to work in areas that I didn't know much about before, and kind of starting from scratch, learning and imaging using MRI was one of these areas. And yeah, I started by working with collaborators from NIH, Danny Reich, and also from Johns Hopkins, Dr. Calabresi, and they introduced me to clinical imaging. And at the time, I didn't know much about it, and I still remember, I'm not sure if you remember that, but at some point Danny gave me a file. That was my first exposure to imaging, and that file was dynamic contrast enhancement magnetic resonance imaging, DCMRI. It's a fancy way of saying, a person is injected with a substance, and then you see how that substance moves and changes in the brain, right? Essentially looking for leakage of blood in the brain, right? So, and I went to many presentations, and I said, okay, I'm going to start looking at it. And little did I know that I couldn't open the file. That was my first. That was
back before R had anything to. Yeah.
And I went to the best person I knew, and that was you. And we tried to open it, and we were able to open it after about 2 weeks of work, and that was actually both humbling and inspiring, I think, to be able to do that. And in the end, what I did, I essentially just bought more RAM for the computer, and that solved it.
Yeah, that's right. Yeah. So there were all those problems then. You had byte swapping problems, and then you had at that time, if you tried to read in the whole image, you ran out of RAM, just one image.
And that's how I also learned that not all imaging is the same, right? So it was just this large file at the time was 1 gigabyte, and everything was freezing, and then you learn that MRI has this very high resolution, and then if you have time series of that, it's even bigger. So, and yeah, that led me to looking into clinical imaging, and we looked into especially lesions in MS, and in the dynamics of the lesions over time, and we found many interesting things. So it also led to many methodological developments with my collaborators, with Taki Shinohara, who built this wonderful group at Penn, with John Mushelli, who's here at Hopkins, and with Elizabeth Sweeney, just to name a few of the people who have. And it's a work of passion. It's beautiful, right? So you open up an image and you see inside the brain. It's what keeps me going.
It's interesting too. You've devoted probably more time than most biostats people in what I would describe as the kind of earlier stages of the process. If you go from kind of raw scanner to sort of analytic structure. I try as much as I can to stay as later in the process as I can. I'm wondering if you can comment a little about the work you did on processing, which is it's more kind of typically lives in engineering departments more often than it lives in biostats.
Yeah, and it's been so to make it clear what we have done. Essentially, we were looking at imaging, and I just want to make clear where we started with this and how we. So we were interested in the brain of people who have MS, and we had images that were taken, say, a few months apart.
MS being multiple sclerosis.
Yeah. And we wanted to know whether something changed, whether lesions got bigger, whether new lesions occurred, whether some lesions resolved or disappeared. And it was pretty clear. You take two images, right? 6 months apart. You register them, meaning you put them in the same space. Subtract the two things, and if it's zero, there is no change. If you see something, there is change. Our problem was that as we were doing that and we were taking these differences, we were getting not zero. And then we realized that something fundamental is going on, and that's how we started to think about, well, if you take these images and you expect no difference, then you should get zero, and why don't you get zero? So it was that fundamental, and then that led to a lot of things, including image normalization, meaning that numbers should mean roughly the same things in images, and then it was followed up by work done by Taki Shinohara. Mainly, I was just a cheerleader on that, on essentially putting images from different studies together, and that I think that work that Taki did create a revolution in imaging, especially because of new studies that are multi-center, multi-scanner. How do you put these things together from different things and make sure that you don't only see scanner effects and batch effects? And that led to a lot of interesting work.
Yeah, it's nice. I never thought about it that way, but I guess what you were trying to do was establish a measurement in the sense that Celsius is
exactly.
Trying to establish a unit for the.
It was as fundamental as that.
Yeah, you wanted to subtract numbers, and they didn't have the same units. Yeah.
Yeah, I mean, if you want to do regression, if you want to do machine learning on these things, whatever, artificial intelligence, whatever it is that you want to do, well, in the end, if you cannot subtract 1 minus 1 and get 0, there is no way to do more than that, which is regression and all these other things, right? So, and yeah, that the rest is history.
Awesome. I'm wondering, and you managed to kind of couple your interest in neuroimaging and MRI with this interest in functional data analysis. So I'm thinking of, for example, MFPCA, and I don't know if you talk a little bit about some of these things.
Yeah, so at the time, you know that very well, right? So one thing that was very important and continues to be very important, will continue to be very important, is studies where you observe people over time, right? And this is one of the fundamental things in biostatistics in general, it's part of our culture here in the department. Longitudinal data analysis, right? You start with a group of people and you observe them over time. So nothing new there, right? But what was new was new tech, that all of a sudden you can get people's imaging over time. You can get their physical activity over time. You can get many other things over time. And these are not, none of this is one data point, right? You observe an entire image and then you observe it again and you observe it again. And that requires a set of tools that we already had, but where the fundamental observation is this very high dimensional observation, and you had issues with how do you compute, because you can write probably the models in the same way you write models for standard longitudinal and multi-level models. The problem is, do they compute? And if they compute, do they give you reasonable results? And that's where we started, and that's why we started working together on multi-level principal component analysis and longitudinal principal component analysis. And then we run into problems of high dimensionality, and again, I was often humbled, and then we tried to do a little bit of work to move the goalpost a little bit farther.
So switching gears one more time, your most recent work is actually migrated away from, not migrated, but included and maybe focused a little bit more on what you might call wearable and implantable technology.
Yeah.
Anyway, can you tell us what wearable and implantable technology is, why it's interesting, and then maybe you have some controversial takes in that area, and we'd like to get to those.
Yeah. So briefly, how do you change to something new, right? How do you? So I was lucky, and this time again, I can blame you for that because Tom Glass approached you.
Yep.
And he was doing some work on recognition of movement from accelerometry, and I think you sent him to me where we met.
Yeah.
And then we started working on that. So what is wearable and implantable technology? Well, there are all these new gadgets, devices that we wear all the time, and they measure different things. For example, an accelerometer on the hand measures the gravitational units, the acceleration of the wrist during the day. You can have a device on the heart or on the hand that measures, the electrocardiogram. You can have an implantable, because, an implantable could be something as simple as CGM, continuous glucose monitor. It could be more implantable, like a heart pacer or something like that, depending on the level of technology. But we started with something simple. We said, okay, this is a new technology, new technologies, in fact, many, many new technologies. And we wanted to know whether they make any difference. We thought they might. And we thought we are getting to better, more explicit measurement of what we were trying to get. And at the time, we were very interested, and we still are, in making the connection between this new technology and seeing whether or not that has anything to do with health. And that was the idea.
Does it have anything to do with health? Is it?
you know my opinion on that. So, but I will. Yeah, absolutely. And this was probably one of the biggest surprises for me. Is just, and I'm going to take just one example: accelerometers, which measure essentially how much you move your hands or how much you move your body. And we asked very simple questions. If you put such an accelerometer on a person, is that related to their probability of dying within the next 5 years, and or developing a new disease such as multiple sclerosis, which we talked about, or Parkinson's disease, and the short summary of that is that we find again and again and again that physical activity as measured by these devices objectively is the best predictor for incident mortality in the US population, in the UK population, and we find it is one of the best modifiable risk predictors for neurological diseases such as Parkinson's and Alzheimer's. And it's just surprising every time we ask the question, how about this other thing? We find again, yes, it is the strongest predictor for that.
And how far in advance are you going? Like, is it 5-year mortality? Is it 10-year mortality?
It's all of the above. It depends on the study and depends. So for example, with NHANES, we can go up to 10, 12 years. And we continue to accumulate data because these were studies that were done in 2003, 2004. So we accumulate the data. UK Biobank right now is 3 to 5 years follow-up, but it will continue. So, and all indications, especially for mortality, seem to be that physical activity is the strongest predictor, especially for immediate mortality, for 1 to 5 years. And then the signal starts to get a little bit weaker, and then other predictors such as age take over, which makes perfect sense.
Yeah. Now, and do you think this will progress into the idea that activity is an intervention? So you know that you could say, okay, if you ramp up your activity by however many,
right,
10 units or whatever, you're decreasing your mortality risk by X, and we're gonna treat this as a drug in the same way some kind of preventative medication would be, or something.
Yeah,
like that.
we haven't answered that question. I mean, to be fair, what we showed is that physical activity predicts mortality. So we haven't shown that, which is harder, right? So intervention studies are hard, difficult. They require a lot of additional things. Everything we have done seems to point out that that's an interesting target for intervention, probably one of the best targets for interventions.
Yeah, we're certainly targeting worse things for intervention. Yeah,
exactly.
Things with less indirect evidence. Yeah.
And I think that clinical trials and others, especially when you have rare outcomes, right, like mortality. If you have a study of 1,000 people, which is kind of standard for a clinical trial, you don't have a lot of mortality in the study, which is good.
Yeah.
But then what do you target, right? You want to target something that is very predictive of mortality. So I think that this is where we are going. So the answer to your question is what I believe. I think that it's a fantastic target for further study, and especially probably one of the best targets for for intervention. It's modifiable. Now, how much of it is modifiable, we don't know, right? What portion of this is modifiable? Then there are all these factors related to culture, to environment, and many other things that, if you don't have a place to walk, then how are you going to walk, right? So it's a complex.
Have you differentiated different kinds of activity? Just that you mentioned walking, is there? First of all, can you detect in some of these studies different from the accelerometer different kinds of activities, and then are the different kinds of activities more or less related to mortality or whatever?
It's ongoing work. It's ongoing work, and we are, I would say, a lower resource environment than, say, Google or Apple or others who already have their own methods and algorithms to detect, say, walking, right? So what we are doing, we are taking open source software, people who said that they can estimate walking, and then we look into whether those algorithms, first of all, do they give reasonable results in terms of number of steps, in terms of these things? Do they agree with each other? And is walking more predictive than, say, overall movement of the hand? But yeah, we are moving in that direction. We don't have definitive answers right now, but it seems like walking is an important part of the discussion. I think that sedentary time, which is essentially what we're doing now, is probably an important part of the discussion. So there are things that are emerging as important.
I should sidebar this too, is that now if you meet with Ciprian now, it's a walking meeting. So there is an intervention actually going on in the Department of Biostatistics now that are kind of floors on a loop. And if you meet with Ciprian, it's now a walking meeting. So since you've been working on this, it kind of went, as far as I can tell, from, there was relatively few people working in this area, and many of the studies were, I guess, what I would describe as lab studies with accelerometers like attached to everything, to now a lot more passive accelerometry but also much bigger studies. And it seems like now any big cohort study or something like that is now collecting. Is that how it is now?
It's changing dramatically, of course, and I think that there were a few studies that did it really well, or they tried to do it according to the best standards of the time, and that includes NHANES and UK Biobank. So in UK Biobank, we have up to 100,000 people with 7 days of accelerometry data. That data is just absolutely enormous, and so now you have 100,000 people who at some point you have their movement, and then you see what's going on after that. So it's definitely going in that direction. We are trying to work with collaborators and explain to them why this is important, and many studies have started to collect very high quality data. We are also learning about how to improve the data acquisition and how to enrich the passive collection of the data with contextual data. For example, asking people using a phone app, simple things like, "Are you happy now?" Right? What is your level of energy? How energetic do you feel? And kind of getting this physical activity in the context and relationship with human perception of what's going on, and that really adds a new dimension to these studies, and then Vadim Zipunikov in particular has looked into this relationship between mental health and physical health. And the good thing about this is that we are now targeting, I think, what we always thought are the correct targets, which is physical health and mental health, and we try to get objective measures of that, or as objective as possible, and that was not possible before.
And is there anything? Imagine you had an older study that collected a lot of stuff, mm-hmm. but didn't collect accelerometry. Is there anything that, I guess, other than mortality, is there anything that you could use as a surrogate for physical activity? Is questionnaire physical activity a reasonable surrogate? Or do you find, is it like nutrition, where if you ask people what they eat, it's very different than what they actually eat? Or is it, are there other things that can be used as proxies, I guess?
Yeah, so, the short answer, the surprising answer to me was that objectively measure physical activity and subjectively measure physical activity are very uncorrelated. And I expected that correlation to be high. Now, that's good and bad. That's good and bad. We don't know exactly what, but I think it's good and bad. So the bad news is that they are not correlated. The good news is that they are not correlated. So what do I mean by that? Well, if you have one's perception of their physical activity and their objective measure of their physical activity, and they are not correlated, but they both predict mortality, then it means that you are getting at two different things, right? One is one's perception of physical activity, and the other one is the objective part. So that's good, right? The bad news is that what we thought we were measuring is not what we did measure, right? So the interpretation, the translation of that is very different. So that's what we're working with. So we find that the correlation between self-reported physical activity and physical activity is very low. The same thing is true for, say, things like sleep quality and all of this. It's just human, the way we ask the question, the way we get the answers, and also the resolution, right, at which you can measure things. With the device, you can measure things at sub-second intervals. You cannot ask me simple things like, when did you walk 2 days ago?
Yeah.
At what time did you? I don't know, right?
Yeah.
I think I was active, but how active was I? Right?
And how, so like I use my little walker thing on my phone.
Mm-hmm.
Is how accurate do you think that is? Is it good? When it tells me I walked 3 miles in a day, I'm like, oh, good, I walked 3 miles. Is it possible I walked like a mile?
it is possible, but these devices are getting better.
Yeah, and they use the GPS too, I assume.
Yeah. So again, these devices are based on technology and amounts of money that we don't have access to. They are also, closed. They are not open source, and I cannot verify exactly what they do.
But in the studies, they're all the like NHANES or whatever. They're using some. They're not using passive phone monitoring. They're using like some.
They're not, but some studies that I'm aware of are using phones,
watches, phones, yeah.
and that leads again to the same problem that we've had with imaging, because it's not clear that phone steps, when you have your phone on you, is the same thing as activity counts measured by a wrist-worn device. And so, and that's where some of our work is going towards this idea of, if in 10,000 years from now somebody wants to look at our studies, what is it that we can do now that would be unchangeable 10,000 years from now, right? And in some sense,
it's a pretty long horizon.
It's a pretty long horizon, but suppose that I'm asking this kind of extreme question, right?
Yeah.
And then you realize that, acceleration is acceleration. That's not going to change. And if you measure acceleration at a reasonably high resolution, and you keep that data, that shouldn't change that much. Maybe the accelerators will get a little bit better, maybe, but essentially that wouldn't change that much. And that's the idea, and that's what we are trying to do with many of the studies, to keep the raw accelerometry data so that we can go back to it and make sure that the numbers from one studies are translatable directly into the numbers of another study. I hope that makes sense.
Awesome. So what kinds of new stuff are you working on? What are you excited about now?
I'm excited about just about anything. I'm inspired by what you do, which is you're working on things that are always cutting edge, and but this is the beauty of my work, right? And not of my personal work, but of my job, is that I walk in here, I never know what's in the box of chocolates. I never know, right? And one day, I started just about a year ago. I started working with collaborators from the surgery department here at Hopkins, and they are interested in cardiac surgery, right? right? And we went and saw the cardiac surgery. Some of the students went with me. We dressed up. We went in there to understand what's going on, and it's very interesting. Essentially, we work with collaborators who work on transplant surgery, on heart transplant surgery, on bypass surgery, and their questions are simple yet complicated, right? All surgeries are very well monitored. Data are extracted from basically every second the person is there for up to 8 hours or more, depending on how difficult the surgery is. And the fundamental question is how to manage their blood pressure, how to make sure that things are managed as well as possible during surgery. And all of this data essentially is stored, and we are asking questions of the type: who develops adverse effects and for what reasons, given all this data that we now have access to? And it's an incredibly complicated problem. We are looking at things like, is there enough blood going to the kidney? Is there enough blood going to the brain? How do you measure that? And these are extremely interesting open problems that we are working on.
Nice. So it was a caramel in that chocolate. It was a good one. Yeah, it wasn't like that. You bit into it. Wasn't like a cherry or something. Like the bad chocolate in the. Yeah. But that's awesome. I find the best collaborators to work with are the ones who ask simple questions and have a kind of innate quantitative sense, but maybe don't know a lot of the methods. But they for whatever the reason have some kind of quantitative instinct.
Yeah. Absolutely. Yeah. I mean, it's I think in general there is nothing better than kind of trying to do something, right, and then understanding what are the complexities associated with that. Right? So in statistics, I think, when we teach statistics, when we learn statistics, when the data are kind of there, right? So there are, you open up R, here are 20 data sets, here are the examples, here's how to do a clustering analysis.
The iris data.
The iris data, and that's fantastic. You learn all of these techniques. My reality is a little different. You start with a question that is more or less well defined, with
typically less,
and then you try to go from there and see if you have the right data, whether you can collect different data sets, and more often than not, you realize that you have to spend a serious amount of time just cleaning up the data, understanding the structure, and understanding the complexities. Like, for example, in surgery, not all surgeries are of the same length, right? How do you compare time in one surgery versus another? Not all surgeries are of the same complexity. So, learning these things. We were just looking at measuring blood in the brain, essentially using some sort of spectroscopy, infrared spectroscopy. You have to understand what is that, right? So essentially you use lasers and you apply something here, and then you try to get the amount of hemoglobin in the blood. Now, how good is that? How do you know how good that is? Right? So, and I asked my collaborators, well, can you put it on a watermelon? Let's put it on a watermelon. There is no
put the freaking lasers on the watermelon, man. Yeah.
And they did.
And they did. They really did. Yeah. Okay.
It was 50% hemoglobin there. So but that's part of just learning and being humble about that. And realizing that you don't know not anything. You don't know everything, and there is much to learn from your collaborators.
This sounds very nicely clinical too. This topic too. That's always really fun. So I have some rapid fire questions. We're hitting toward the end, and I don't want to take up too much of your time. So I have some rapid fire questions, completely unrelated. Most of them completely unrelated to statistics. Favorite place to eat in Baltimore? Favorite place to eat?
Favorite Zorba.
Zorba. Okay. Best place to get calamari in the world? Because so Ciprian and I have had a long-standing discussion over where you can get the best calamari, and we've had Ciprian's quite opinionated about this. So best place to get calamari, if you had to give it anywhere in the world that you've been.
Unfortunately, the place is closed, but I'm gonna name it. It's Pizzicato in Philadelphia.
Oh,
Market
they
and
closed.
3rd. They closed, unfortunately.
Yeah.
I think that the place we went to in South Africa was
That
pretty
place
good.
was amazing. Yeah.
Was pretty good. But there are a few good places here in Baltimore. Black Olive would be one of them, I would say.
Nice. Is Meze still open? Is it?
Meze? No, they closed.
Oh, they closed. Oh.
Yeah. But
they had great
Zorba,
calamari.
again, have good calamari. It's just I would add a little bit of lemon.
Awesome. And so you just got back from some travel. What are some of your favorite places to travel to?
I love to explore the world. I've been to many places. I went to Turkey right now, and it was absolutely fantastic. And then I went back to Romania with my daughters. They love to learn new words, and I found out that they're learning all the bad words first. But they like to kind of immerse themselves in the culture, and it's interesting to see them switch between being American here and trying to pass as a Romanian in Romania. And it works, except for some of the verb conjugation and some of but it works. It mainly works.
So in case you didn't notice from the number of vowels in Ciprian's name, Crainiceanu's last name, he's Romanian. So if you had to say, if someone's visiting Romania, where do they go? Where should they go?
You should talk to me about that.
Yeah.
It depends. It changes a little bit, so it depends on what you want to see.
You're a beachy person. Let's say you're a beachy person.
So you could go to. It depends on what kind of beach. If you prefer the Ocean City type beach, then probably Mamaia, but if you prefer more kind of the isolated end of the world type thing, probably Portița or Sulina. These are kind of end of the world beaches where nobody bothers you.
You'll be all alone.
You'll be all alone.
Awesome.
Yeah.
Awesome. Great. Well, we are almost exactly at an hour. Do you anything else to add? Anything you'd like to bring? Anything to pitch or anything like that?
No, I'm very happy. I hope that I provide a little bit of a window in what I'm doing, and I just wanted to say thank you for everything you are doing and everything you have done for me in my career.
Same, likewise, yeah. And thank you for coming on the podcast.
Absolutely.
Yeah, so in case you didn't know, the way we get guests for this podcast is usually, like, we record on Tuesdays, and it's usually Monday evening. So thank you for saying yes on late notice. And it's been a pleasure working with you, and my best collaborator. So thanks once again.
Thank you, Brian.